Three, two, one, go. Hi, everyone. Welcome to this really interesting episode, which I've been looking forward to for a very long time. I've got a really special guest today. I've got Dr. Bhaskar Mukherjee. I don't know how to start describing about him, but I think anyone who's worked in psychiatry in India at this point knows him and really respects him. He is considered to be a thought leader as far as the neurobiology side of things is concerned, and ever since I was in residency, I've idolized him and looked up to him to attend his lectures, to try and identify as to how and where to start picking the pieces up of our very complicated brain. Thank you, Dr. Mukherjee, for taking the time out to do this today.
It's my pleasure actually, and thank you for calling me here because I love to talk about brain and I love to make people educated about brain and understand that how and where this complex mechanism that controls us gets disturbed and create the various diseases that we don't see. They are the invisible diseases of humankind. They are the origin of every non-communicable disease, but they are relegated to a garbage bin and called as diseases of mind, disease which don't exist, and various other things. So, thank you for calling me here, and thank you for letting me explain to people that how these diseases actually destroy humanity and are going to be the major cause of death once we conquer the infectious diseases.
I think it becomes even more pertinent with the most of the, not only the Western countries, but even our subcontinental countries appreciating the mental health epidemic and identifying that there's a lot of need for awareness being created. Were you always, during your MBBS days, were you always interested in psychiatry? And was this something that you always wanted to pursue as a profession?
Mostly, I am interested in brain. I see brain. I don't see psychiatry or I don't see neurology. I don't see it that way. I see it as a whole brain. I was interested in brain because, let me be very clear, heart is mostly discovered. Liver is mostly discovered. We know most of the gross things about them. Yes, there is a invisible realm of molecular biology. There is an invisible realm of bioelectrical field that we don't know about, but other than that we mostly have figured out other organs. But what makes us human, what makes us animals, the brain, the spinal cord, we don't know anything about it. Even after all my life trying to learn about these things, I would say I know 1 to maximum 3% of what needs to be known in brain. So that is why I was interested in knowing brain. Then that's how I ended up in psychiatry.
So you were always very clear that it's the neuroanatomy side of things and the neuropathology side of things that fascinates you.
Yes, neuroanatomy, neuropathology, neurogenetics, ultimately the building block of life.
Just to get us started, like we discussed before we started shooting, that this podcast, most of the listeners are people who are interested in mental health. A lot of them are mental health professionals. And we discussed a starting point because we usually do want to deep dive and we want to go into excruciating details and we don't shy away from it. So we agreed that I think as far as the starting point of this discussion is concerned would be that if people know the basic lobar functions of the brain, I think that would be a good working knowledge point of view to start.
Yes.
I'll try and share my screen when we eventually edit the video so that helps a visual spatial sketchpad of sorts for people to sort of grasp the thing as well. But to get us started, talk to us about just some basic working knowledge of the brain which you would want people to know before we deep dive.
Yes. The basic I would talk about mostly brain from a evolutionary perspective of think fast so that people can understand what is brain. Let me start with a unicellular organism. When we talk about unicellular organism, they have just cell surface receptor which catches the external environment and its changes, then those cell surface receptor carries those information through intercellular pathway to nucleus, and nucleus then causes various changes in some other intercellular pathway, which causes ultimately flagellar movement, cytoplasmic movement, and various other things, and makes that unicellular organism go in different evolutionary adaptive mode or different environmental adaptive mode, whatever we like to call it. Now, this is how a unicellular organism like amoeba works. But when this unicellular organism starts growing and becomes a multicellular organism, then this kind of adjustment which in the unicellular organism does cannot be done, because let's say one part of the polycellular organism or multicellular organism is exposed to heat. We don't know where is the central point. So that exposure to heat has to be conveyed to that central point. Then that central point has to take some decision, and ultimately that is why specialized information carrying cell has evolved. These cells were primitive nerve cells, and in animal world it is first seen in anemone, hydra, this type of multicellular organism who has a nerve net. This nerve net connect one side of body to other side of body, and through this nerve net they control their expressions and reaction to surrounding environmental change. But as the multicellular organism grows, its various aspects of interaction with external environment also grows. Let's say we first need to control the pH in an organism, then metabolic demand, then temperature, then various water and other balance. Ultimately, it becomes quite a tedious process where multiple nerve loops controlling each aspect of the internal environment has to adjust, readjust, and continuously a dynamic adjust to create a perfect living environment inside the organism. This created multiple loops of nerves and multiple centers of adjustment, and this is the primitive CNS. This part is seen in cephalopod, let's say octopus, let's say squid. In octopus, squid, there are multiple such nerve loops which control their every aspect of internal environment and changes this dynamically to response to various external environmental change. Now, beyond the cephalopods, it became clear to evolution and nature that this type of segregated nerve loop cannot give an animal enough evolutionary advantage to make it evolve further. So, for the purpose of evolution and for the purpose of making things simpler, because nature is something which doesn't waste energy. Nature is a very strong advocate and proponent of thermodynamics. Nature doesn't waste energy. So nature has to create something that would bring all these central regulators together. This started in those animals who were first amphibians, and in them the various centers connected together to form primitive notochord and primitive CNS. That is how central nervous system journey started. Then onwards, amphibian onwards, when reptiles came, when mammals came, they started living in a cohesive family environment. And so, along with external environment, physical environment, there came a layer of social environment, where the animal has to instantaneously respond to the social need of other similar type of environment. Let's take a family of elephant. Let's take a family of primitive dinosaurs. All of them needed to respond to physical environment and primitive social environment. So the micro adjustment between various regulatory center had to be more and more complex, and that gave rise to neocortex. Neocortex is that part which connects one part of regulatory system to another part of other regulatory system. It connects temperature center to metabolic center to osmotic center to other things. And as our external environment, our social environment grew, the animal's neocortex also grew, and ultimately it became the hugely developed neocortex of anthropoid ape and humans. That is how brain evolved. So if we talk this way of evolutionary look at brain, we would understand that brain is the headquarter and everything where animals internal body homeostasis is controlled, and that is what brain is. I hope this part has been cleared.
Absolutely. Fascinating introduction to, and I think a backdrop to the conversation that we are now going to move into. Please, happy to follow your lead. What's the next chapter then?
So if by evolutionary journey we have reached to a complex primate who is adjusting continuously to its own social environment, then its own physical environment, and making change inside the body to suit to it, we get some idea of how human brain works. And then starting there, we need to go deeper into various processes that lends brain the power to continuously micromanage the various aspects of body, the various aspects of body function, and the various aspects of expression of brain, which ultimately results in meaningful social communication, meaningful physical communication with physical environment, and ultimately leads the human being which is trying to lead a meaningful life inside a very complex technological world that we live in. So that is how brain is operating, and for knowing that, we need to know molecular neuroscience, we need to know connectomics and connection neuroscience, and we need to know various systemic neuroscience, where how one system is interacting with other. So this is the backdrop I would like people to understand, and from here we can go on based on what you want to know. But this is the basic
Fabulous.
of brain.
So there would be 3 specific things that we would dwell into if we discuss any sort of function or pathology, and one of them is the molecular neuroscience. The second one is the systemic, or how these molecules are interconnected, or these structures are interconnected, and the study of the connections as well. So the structure of the tissue itself, the structure of the connections, and how they work as systems. These would be 3 broad strokes. Let's deep dive into a basic idea about the brain, the normal functioning as far as the psychosis side of things is concerned, and then what leads to development of delusions, hallucinations, or even negative symptoms.
Yes. So let's first talk about what is normalcy, because that problem in psychiatry is we don't talk about normalcy, we don't really talk about pathogenesis. The problem is pathology in case of brain is intermingled with normalcy. Until and unless we describe how brain normally functions, we cannot understand what is abnormal here, because every attribute of brain that we are talking about is normally distributed. It is present in everyone. Each of us are slightly obsessive, slightly attention deficit where things doesn't concern us, slightly paranoid when we feel threatened, slightly anxious when external environment or our internal environment overwhelms us. So all of the things we describe as pathologies are actually part of a normative spectrum that is present inside us. So standing there, we need to know how normally brain functions. So how normally brain functions? Brain is a primarily information carrying, processing and responding organ. So in any case, in any scenario where brain is just doing it normal day-to-day maintenance work, brain normally carries the information that it is subjected to, and based on the information processing and information interpretation, it responds. But whenever, let's say, brain gets something out of ordinary, let's say change of weather, this is a very normal daily occurrence, change of temperature or change of weather, whatever we may call it. For brain, it is a challenge because brain was working in a different temperature and suddenly it is facing heat or cold. So it is a challenge. Now how brain would react to this challenge? First, brain would pick up the temperature change from peripheral sensory neurons. Then the peripheral sensory neuron sensation would be processed in brain, localized, and would be compared in different past experiences of brain. Means, let's say, for some brain, a warm temperature is more soothing. For some brain, a colder temperature is more soothing depending on how the brain functions, how the different past experiences and past conditioning were. Now, based on that, brain would start trying to create a new balance. For that, various areas of brain would start working more and more. Whenever brain is working more, there would be activation of various intracellular pathways, and those intracellular pathways and new metabolic need of that pathways would create more mitochondrial energy. Now mitochondrial energy production would create, number one, more energy for every neuronal cell, but along with that it would also produce various metabolically active reactive oxygen species, as well as it would produce a energy demand on brain cells and total brain state. Then this is one side. Then in another side, there would be more and more protein turnover in brain when brain cells are hyperactive. So for protein turnover, new proteins have to be synthesized. For that synthesis, various transcriptome activity would get enhanced, and for that
Sorry, I lost you, Dr. Bhaskar. Hello?
at present,
Yeah, we're back. Yeah.
actually that was basically my screen mirroring is on, so
Okay.
a phone came in my iPhone and anything. Anyway, let's go
No.
back.
Yeah.
So how till how long we are connected?
So we said that it would need more protein because the mitochondrial energy generation would increase. It would lead to increased energy demand, and that would lead to more protein
No,
requirement.
that would lead to reactive oxygen species generation, and that would lead to a lot of reactive oxygen species mediated cellular transmission and cellular stress. That is one part. Then another part is, as cells are hyperactive, we are going to need more and more protein for maintaining this hyperactivity. So cellular protein recycling would increase. This would lead to transcriptomic activation, and transcriptomes would produce more and more protein. And while we are producing protein, this protein would fold amongst their internal superstructure to produce various new structure, and amongst these new structures there would be various proteins who don't have a intrinsic stable structure, the intrinsically disordered protein. They would actually form backbone of various processes, but when protein production is hyperactive, these proteins and their internally disordered region, they frequently misfold and create various wrong superstructures which needs to be removed. That produce a stress on cellular protein production mechanism as well as a stress on cellular protein digestion mechanism or ubiquitin proteasome system. This is known as misfolded protein response or various proteostatic stress. You can call both. Both are partially correct. None is true, the real name. We haven't really found a actual defining name for it. This is called intrinsically disordered protein response. This would create another side of cellular stress. Now the third is, due to these two, there is also endoplasmic reticulum stress. So there are three processes of cellular stress: mitochondria through its reactive oxygen species generation, then intrinsically disordered protein response, and endoplasmic reticulum stress. These cellular stress which are produced would be sensed by various intracellular pattern recognition receptors. Pattern recognition receptors are receptors which actually recognizes various molecular structure and various molecular signals inside cell, and when a specific type of molecular signal is produced, they pick it up and they activate the stress sensing mechanism. The stress sensing mechanism can be activated due to virus, can be activated due to increased cellular stress, can be activated due to a lot of reasons. But these receptors are known as pattern recognition receptor. There are some name we know, most people know, toll-like receptor. Some names most people don't know, like they are known as rig-like receptor, node-like receptor, and other things. They get activated when this type of cellular stress happen, and through their activation, they activate various heat-sensitive protein. Then there is a specific pathway known as MyD88 pathway which gets activated, and ultimately there is damage associated molecular pattern, and that response, which is known as damage associated molecular pattern response or DAMP response. DAMP response signature, when secreted by cell, that is picked up by immune cells, that is picked up by various innate immune system component, and that creates initially growth, because innate immune system is also growth system. So initially innate immunity stimulation would create growth signaling, and that growth signaling would start producing various growth results. This is the first step. I hope so far I have been lucid enough. Yes.
Absolutely.
So this is the first step. So in first step, we discussed how brain cells hyperactivity creates cellular stress, and that cellular stress create changed immune signature of cell, and that changed immune signature would produce various growth signals and start causing various extracellular change. Now, what would be the extracellular change? The first extracellular change that would happen is there would be activation of microglia. Microglia is a cerebral residual immune system cell which is paramount in brain repairing, brain defense, brain growth, and various neural microadjustments, which we grossly called neuroplasticity. Now, microglia stays in three states. Normal is MEM0, which is neutral. Then when it is active and is first it is pro-growth, then pro-inflammatory, then it is called M1, and then which is anti-growth and anti-inflammatory is called M2. Microglia transits between M0, M1 and M2 depending on various innate immune system responses. Amongst them, one is this damp signaling. So, whenever there is damp signaling, microglia transits from M0 to M1. And due to this transition, M1 microglia starts activating various areas of brain and starts creating various changes. It first activates astrocyte. Now, astrocyte activation would again lead to astrocyte changing it from A0 to A1. And that A0 to A1 change first would try to inhibit microglia and second would try to activate the blood-brain barrier, because astrocyte input is actually one of the component of blood-brain barrier. So whenever astrocyte is activated, through astrocyte, blood-brain barrier cells are activated and astrocyte causes activation of the vascular endothelium as well as the vascular pericyte that is wrapping around the endothelium. That would lead to ultimately activation of neurovascular unit, and there would be coupling of neurovascular response. Means with astrocyte activation, there would be activation of blood vessels, there would be vasodilatation, there would be more and more spreading vasodilation, leading to more metabolically active blood reaching to brain and meeting metabolic demand of brain. This term, this neurovascular coupling, if you go into various literature, you would find that neurovascular coupling and its maladjustment is part of various abnormal brain responses, be it autism symptom, be it psychosis, be it chronic bipolar, be it neurodegenerative disorder. Everywhere this neurovascular coupling and its disturbance is listed as one of the causes. So that is how we are linking the pathology with physiology. Neurovascular coupling is a physiological phenomenon, but its disturbance would lead to various pathological expression. Now, along with that, activated microglia would also cause activation of inflammasome. Inflammasome is actually a complicated machinery which causes propagation of inflammation inside body. Inflammasomes are present throughout the body, but inside the brain they are activated by astrocyte as well as microglia, and those inflammasome are known as NLRP3 inflammasome, not like receptor 3 inflammasome. Now this NLRP3 inflammasome would propagate the various inflammation throughout the brain and cause propagation of this growth response. Initially you have to understand that what we call inflammation is extreme state of growth response. Initially growth response is beneficial for our brain, but when growth signaling becomes more intense, it becomes pathological, and that is a very fine line. So this is the way neurovascular coupling would happen. Then this activated microglia would go into various neural proliferation zone. Now this is another interesting area that needs to be understood. When you or I were MBBS student, the standard dictum was brain doesn't grow. Brain grows all it can grow between 16 to 18 years, and then it can only die and things like that. that was like kind of like inevitable date for brain and things like that. But in last 10 to 15 years, we came to understand that this is not such a grim picture. Brain has multiple areas where there are brain's own stem cell needs, and these areas continuously produce new cell when it is required. And one of the strongest mediator of new cell production is activated microglia, M1 microglia. These stem cell needs are in hippocampus, which is mostly well known by now, but the largest reservoir are inside the ventricular wall and subventricular layer of a brain. So these areas are activated by macroglia, and these stem cells start producing new daughter cells, which are then transported to the hyperactive areas of brain, which were at this moment hyperactive to manage the environmental change, that is just change of temperature. So these cells would go to those hyperactive areas, would settle down there, and would produce new synaptic connection, and brain would start adjusting to new state of brain, which is more information carrying, more functionally active, and brain would overcome this challenge. This is how physiologically brain works. I have just given example of temperature change. The example can be anything. The example can be a post-viral illness. The prolonged malaise of post-viral illness is actually brain adjusting to the new reality. The grief reaction, it is also the same thing. Brain adjusting to new social reality. And so it takes instantaneously for temperature regulation, but for complicated bodily state and complicated social and environmental states, it might need up to 6 to 8 weeks. That is why it is said that grief lasts for 6 to 8 weeks for most people. There is a actual scientific and physiological basis for it. So this is how brain normally physiologically functions.
Perfect. And would you say that the last bit is also something which is similar to what we would in common terms call neuroplasticity, or would that be something completely different?
No. This is one of the major areas of neuroplasticity. Once
Perfect.
brain starts create new cell, brain starts create new synapses, brain remodels those synapses. That is what neuroplasticity is. Neuroplasticity is actually a lot of stuff. Whole body is plastic. Our whole body is plastic. Most plastic organ in our body is bone marrow and liver. They regenerate completely. But brain has not that much plasticity, but brain is still a reasonable degree of plasticity is there in brain. Brain adjusts to a lot of things, and this process of adjustment is known as plasticity. So brain producing new cell, producing new synapses, and adjusting to new reality by increasing its functional capacity is a definite example of neuroplasticity.
One other follow-up question, Dr. Bhaskar, is just to quickly summarize what we've covered so far is that just like any other part of the body, just say our skin,
yes,
our skin functions normally every time it is in a weather and a home condition where it is used to, it will function normally. But say suddenly the weather changes, it will start either secreting more oil or getting very dry.
Yes.
That's what happens with the brain as well. And just like with the skin, there are compensatory mechanisms which would help the skin cope with a different physiological environment. So is that with the brain, that there would be vasodilatation, increased blood supply, increased protein generation, formation of new cells, formation of new connections, and adaptation to that normal change that would happen. And the same thing in some cases would turn into pathology,
yes,
which I think we are now moving towards. Would you say that just like with the change in temperature or grief, any other stressful factor for a lot of people might be they have a breakup, or they start consuming substances, which is again an external stimulant, even though cannabis is similar to endocannabinoids, or opioids is similar to endogenous opioids, but these are still external stimulants which are not environmental or emotional, but these are actual biochemical external stimulants which can similarly cause a change in the normal physiology. Would that be remotely correct to say?
That would be partially correct to say. I would suggest endocannabinoid, endoopioid, and the various chemicals, I would say keep it separately, because that is a very interesting different thing. We would go there later. But for every other thing you mentioned, that is how things happen.
Okay, perfect. So are we now moving towards the pathology side of things?
Yes.
From what is normal? Okay,
So
perfect.
once we understand that this is normal, it is easier to understand that this happens to all of us. First, normal thing is happening to all of us. We all suffer from grief. We all suffer from negative things happening to our life, and we all feel low during those periods. We all actually withdraw. We all actually try to spend as less energy as possible during those times, and that has been given a unique name, illness behavior, in animals. And that same illness behavior is being exhibited by human beings when they suffer from a prolonged illness and are recovering, or is suffering from a social or environmental negative things. So that problem is, due to our current nomenclature, we term this as depression and start treatment, which is entirely should not be done, because again, this is a physiological response. When it turns pathological, there are some clear-cut brain science for it, and we need to understand those things. But as today we are not talking about obsessive behavior, or we are not talking about various brain states that actually give us indication when and how illness behavior is turning into real brain pathological expression. We are not going into that. We are talking about psychosis today. So in case of psychosis, first we have to understand, again, there are some very common things that we do day to day. We all have our favorite things. We all have our favorite opinions. We all have various opinions, various point of views, various philosophies dominating our day-to-day life. We all develop them. In fact, I remember there is a very famous meme which describes the opinion as a dog. We all create our opinion of that. We nurture that opinion. That dog grows, and sometimes we let the dog grow too much, and the dog finally eats us. So that is a very good corollary of what happens in brain in psychosis. Means we all have our opinion. Our brain is biased. Brain is a very biased organ. Brain is not a very trustworthy organ. It doesn't remember information much. It remembers pattern. It is biased. It favors one pattern more than other, and it has a lot of imperfections built into it, because these imperfections are evolutionary acquired. Now, due to these imperfections, brain cannot act neutrally in every condition. This is normally just our opinion, but there are some brains which are in love with their opinions in everything, and they start giving value to their opinion more and more, and they start giving their opinion more value than what external environmental cue they are getting, social environmental cue they are getting, and ultimately these brains are unidirectional and they are termed as fanatical. This is the so-called biological basis of delusion, because these fanatical brains, when they start believing something, they would start believing in that despite everything that is happening contrary to what they believe, despite everything that they are getting as environmental input in contrary to the opinion they are in love with. And that creates a opinion or belief strong enough that is resistant to any kind of logical intervention and environmental intervention. That is delusion. So our biased brain are more biased in some people, and they start believing in that bias more and more, and ultimately those biased brains, when they are destabilized enough, they would go into those biases so strongly that they are not amenable to any logic, not amenable to any evidence that is in contrary to their belief, however strong the environmental cue is, and they land in delusion. Now, this delusion is the basis of the next dysfunction in psychosis, hallucination. Now, what is hallucination? Hallucination is actually something that is very easily explainable through a combination of this delusion and then sensory perception abnormality. Now, what is sensory perception abnormality? We perceive our surrounding environment through some long-range sensory modality like vision, like the sound, and some short-range sensory modality like smell, like proprioception, like touch, like taste, right? Now, we perceive sensory abnormalities in all those systems. Many a times when we are very alert and hyper vigilant, we many a time feel that something is crawling in our skin. Right? We don't consider it a hallucination. We just consider that unpleasant sensation of something crawling. But that is actually a tactile hallucination, which can also be called as formication. And in elderly, that is the basis of delusional parasitosis. Their biased brain, due to neurodegeneration, is believing that formication as a parasite crawling inside their skin. Now, in case of addict, this same formication is perceived as cocaine bug. This same formication is perceived as methamphetamine mite or meth mite and things like that. Similarly, all our close range sensations are full of biases. Many patients with anxiety would come to us and say they are having vertigo, but there is no defect in their any balancing organ, means there is no defect in eye, there is no defect in ear, and we would be perplexed to understand why they are complaining of vertigo. That is nothing but proprioception having hallucinatory behavior. Now the thing is, the same thing happens with our long range sensory or system. Long range sensory system means the area, the vision and the audio. Now, our visual system is very strongly monitored system in brain. They have multiple checks and balance instilled in visual system. Visual system is also dominated by a lot of sensory different character. Our visual perception has different color in it. Our visual perception has different hues in it. Our visual perception has different illuminations in it, and a visual perception has so much detail in it that it is very hard for visual perception to escape the check and balance system that is inbuilt in visual pathways in a person who has intact vision. So visual sensory aberrations in form of visual hallucination is rare and only can happen when the check and balance system fail and brain is grossly abnormal. That is why in the past there was the dictum that if there is visual hallucination, then there is something grossly wrong in brain. But auditory system, on the other hand, is not so evolved in normal people who are visually normal. In them, auditory system has very less check and balance. Auditory system has only single produced sound to accompany it. That sound source cannot be repeatedly screened for anything erroneous. So there is a lot of chance of error in that auditory system. Now, in these people who have this kind of delusional brain, in them, those auditory system which is already erroneous misperceives many a thing, and those misperceptions gives rise to various auditory hallucinations in combination with the inbuilt delusion producing brain. Now, that is why auditory hallucination is much more, and that is why visual hallucination is rare. Now, if we understand this way, we actually get answer to another question that is actually very interesting for a lot of people who are working in neuroscience. Since long we are asking the question that there is a myth that blindness, congenital blindness, protects people from psychosis. There is a myth. I am not saying it is true. It is a myth. Because in your clinical journey, I am sure you would find people with psychosis who have congenital blindness. We all have seen such type of patients, and in your journey you would also find it. But it is a common misperception that congenital blind people are protected from psychosis because nobody knows. But if you understand this way, you would understand that in congenitally blind people, the visual sense it is not present. So only long range sensation that they have is auditory sense. So auditory sensation takes the place of visual sense and become as complicated as visual sense. It develops lots of dedicated brain area to it. It develops lots of compensatory check and balance system, and it becomes as acute as a vision system in a normal visually seeing people. So for these people, it is very hard for misperception to happen in auditory area. So they would have very less chance of auditory hallucination. That doesn't mean that they won't have psychosis. That would just mean that they would need a lot of wrong thing to happen to develop auditory hallucination. But they would have psychosis on their own, they would have delusional behavior, they would have delusional brain, they would have various delusional behavior, various delusional experiences, but they would not develop auditory hallucination as much as is expected in other population.
Yeah.
Have I been
That makes a lot of sense. Yeah, absolutely.
okay? So this is how delusion and hallucinations are produced. I have actually simplified a lot because the actual mechanism is quite more complicated. It would involve understanding of executive circuit, it would involve understanding of brain's inhibition response response inhibition circuit. It would take a lot of talk and diagram in understanding how motivational system and extended motivational system and reward system works. But I have tried to make it simplified because I don't think most of our viewers are going to be very deep into neuroscience or things like that. So I tried to make it simplified. Now it is your job to give me feedback whether the simplified model was understandable or not.
Absolutely. So far, it was crystal clear, very understandable, and we're actually. I'd love to go a little more depth into the circuits that you said. So the executive function, motivation, reward. Should we go a bit more into it? And
Yes.
happy for you to take the lead to explain a bit more on it. Yeah.
So the thing is, then we have to talk about delusion as an external manifestation of choice determining function of brain, because brain has actually an one very interesting function along with homeostatic function. It creates goal directed movement and achieves that. And any animal's goal directed movement is direct by its ability to make dynamic decision. Its ability to make choices amongst various available options. And for that it needs a functional executive cortex, a functional response inhibition circuit which would pull the brake out of the action whether to do it or not to do it, and a extended reward circuit or motivational circuit that would decide whether to go full throttle in a job or not, how much power to invest in a job, how much force to invest in a job. That is the work of motivational circuit. So these three circuits would be actually involved in making a decision. And these three circuits, although I am telling them circuits, they are large scale brain network, because executive circuit is actually a circuit which connects prefrontal Similarly, hallucination is also a dysfunction of various networks. Besides the sensory discrimination problem, it is actually a huge network that creates a internal image of our external environment, which starts in insula of both side, along with anterior cingulate cortex, along with temporoparietal junction, along with various other prefrontal areas and parietal association areas to create a large scale network that gives us an internal brain representation of our three-dimensional position in our external space. And by actually disturbance of auditory processing and visual processing and various other sensory processing, our brain is getting faulty feedback in formation of this 3D image, and that is producing the hallucination process. Auditory processing is more easy to fool, and so the long range senses, amongst the long range sensations, auditory hallucination is more, and our short range sensations are always easy to fool. That is why in normal state also it is very easy to produce sensory abnormalities in those short range areas. That is why in anxiety it is common to have formication, it is common to have vertigo, it is common to have various dysgeusia, it is common to have various parasmia and other things. Various people who have anxiety or hyperactivation of brain, they would complain that they are having difficult taste in brain, not brain, they are having difficult taste in their tongue, different tastes in their, different smells in their nose, and things like that. These are also actually examples of various sensory processing abnormalities.
Okay, that's a really interesting sort of deep dive into the pathology or the formation of the delusions and hallucinations. So our body, so just to summarize a little bit before we ask the next question and move on to the next one, is that the body has a normal defense mechanism to cope with a stressful or a different stimuli, and has normal physiological ways in which it adapts to various environments.
Yes.
One of these things is the way if we talk about the delusions, it is formed mainly by the executive network, the response inhibition network, and the reward or motivation circuit. These three main circuits will also, just like the skin, again going back to the skin analogy, will have normal ways to function and normal ways in which the executive pathway will say, the executive network will change the way it functions, which if gets pathological, it cannot control the choices. Or if the reward circuit goes overboard or too strong, and it is not under the control of the response inhibition, which would hopefully or probably provide that reality check to us. So that response inhibition is unable to provide that reality check and not able to check the reward or motivation circuit, which leads to those opinions which were otherwise just opinions or beliefs. But that doesn't happen, and the brain becomes a little unilateral or fanatic.
Yes.
And those opinions don't come into the realm of rationalization then, and no matter how much challenge we provide to those opinions or how much contrary evidence we provide to them, they still become firm beliefs or facts in the mind of that person, which we then call as delusions. And going back to first year psychiatry, that definition of delusion of being a false, firm, unshakable belief, I think this is the neurobiological basis of that. Should we deep dive, Dr. Bhaskar, a little into the molecular side of the delusion formations as well?
Yes, and that would be a fascinating realm. Let me say very clearly, physics has a quantum realm. Biology also have a quantum realm, and the quantum realm is molecular biology, because in molecular biology it is a endless, infinite world of possibilities where we can see the workings, but by seeing the working we can understand this is infinite variation and we are dealing with infinite things. So when we talk about molecular neuroscience, we have to understand that we have till now grossly understood very little. I am again saying, do not think that I am talking about this means I know a lot about the brain. In my own estimate, I would say I can talk about 1 to 2% of brain confidently, 2 to 3% more if I slightly go overboard. 5% I can guess, less 10 among the less 90%, 10% I know I don't know, and but less 80% I don't even know I don't know. So that is how much area I don't know. So standing there, do not even think that what I am saying is sacrosanct and written in stone. This is what we know till now. Tomorrow we would know some more. 10 years more we know something more. But the more I am understanding brain, the more I am realize it is an infinite variation in an infinite landscape. Probably we are going to be dealing with a probabilistic model of brain and would never know the actual working of every cell of brain in a particular situation to provide the exact response to a thing. Okay. So with this caveat clear, because I don't want anyone to be in dark, let's start with molecular neuroscience and what happens there. In molecular neuroscience, you have to understand that each cell is a universe in itself, and brain has about 120 billion such universes, and those universes together create what we are. Now, in a single cell, there are grossly three kind of movements going on from molecular point of view. One side of movement is which are pro growth, pro cell propagation, pro cell generation, pro cell development. One side of movement is which is trying to limit the generation, which is trying to limit the cell growth, which is trying to limit the changes, and the third side normally is silent, but these are programmed and they would be activated when the growth or when the degeneration drive is most strong, and they would start either sending the cell to recycling, which is known as autophagy, or permanent death, which is known as apoptosis. So these are the three sides of very simplified metabolic profile of a neuron. So when a neuron, when a psychosis is happening, what is happening inside neuron? First, the neuron in psychosis is prone to hyperactivation. So they frequently go into hyper response, hyperactivation, hyper excitation, and that creates a chronic excitotoxicity in these cells. Now, depending on the genetic load of the individual and the somatic mutation load at any given moment, here when I say genetic load, I say inherited genetic load, and somatic mutation load is different. Now, depending on these two factors, this chronic excitotoxicity would progress and someday would kill the cell. The faster it progresses, the faster it kills the cell, the faster is the chance that there would be a neurodevelopmental process that is going to a degenerative process and ultimately would go through a neurodegenerative pathway. But this is one side of it. Then there is another side. One is excitotoxicity. Then these brains which develop psychosis, they also have few microstructural neuroanatomical defects. In them, a lot of networks are not really well connected. Means, let's say, I give a very crude example. Let's say our prefrontal cortex in conjunction with thalamus is sending extension to basal ganglia. Now, prefrontal cortex is normally sending 100 neuron connection, thalamus is contributing 20, and together they are creating a 120 strong input to basal ganglia. This is just a hypothetical network. This is normally happening. But in case of brains who are prone to psychosis, amongst these 120 neurons, only 50% or less than 50% would reach to basal ganglia. Some would go into some other area. Some would go into synapses amongst themselves, and some would develop endless loops. So ultimately, only amongst the total strength of, let's say, 120, only 30 or 40 neurons would reach basal ganglia and rest would be wasted. So when communicating anything from these two nodes to basal ganglia, the networks needs to be activated and hyperactivated all the time, because it is working in less than optimum physiological condition, because it is normally going to need 120 connection, but now it has only 30 to 40 connection. So these neurons would be constantly activated, and they already have a hyperactivity vulnerability inbuilt in themselves. So they would degenerate faster, and that would create more problem in them. So this is the second vulnerability. First is they have a excitotoxicity vulnerability. Then there is a lot of misdirected synapses in various areas. The third part is glial support. The glial support is much poor in a brain prone to psychosis than normal brain. The microglia is more activated, and that microglia activation occurs in the utero. Means a brain which is going to develop psychosis at certain point in future life, let's say 15 years of age or 30 years of age, that brain's microglia are abnormal when the brain was developing in utero, and that happened because of abnormal maternal fetal interface interaction. In placenta, there is a maternal side, there is a fetal side, and these two sides communicate with each other through various ways. This is known as maternal fetal interaction, and the developing brain's microglia were abnormal during those periods in these people who are going to develop psychosis some part of their life. Similarly, there are various other glial cells, astrocytes, there are various other glials. They are also not behaving well from their intrauterine life. The astrocytes are unable to actually reciprocally inhibit the activated microglia. The push and pull mechanism between various glial network that creates neuronal migration is abnormal. Then glial support and formation of blood-brain barrier is weak in them, and the glial fruit processes which wraps around myelin seed and create myelin seed is also not working. This is another way. I am just going to list the numbers of different vulnerabilities, and I hope you are writing them down so that when you show your presentation, you would show the tabular view. Then there is another thing. The thing is, in these people, the innate immune system is destabilized. The innate immune system and its contribution to cellular activism is very loop-sided in these people. Their innate immune system has multiple vulnerability. In some, the innate immunity is not able to activate properly. In some, the innate immunity is inappropriately activated. For example, I am giving one name of mutation. There is a very interesting genetic syndrome known as Brugada syndrome. In Brugada syndrome, Brugada syndrome is known for long QT syndrome. There are lots of mutation in Brugada syndrome, voltage gated potassium channel, voltage gated calcium channel, and things like that. But when a person has Brugada syndrome, their brain's innate immune system gets inappropriately activated, and that gives rise to inappropriate cellular activation and excitatory, and that in turn gives rise to very erratic brain activation pattern, and ultimately in these patients there is a high chance of psychosis. If you see in literature, Brugada syndrome is associated with high risk of schizophrenia, but why that nobody is sure. This is probably one of the reasons. Then there is another genetic syndrome which is known as plasminogen activator gene mutation, PAIG. Plasminogen activator inhibitor gene. Plasminogen activator inhibitor gene has 4G mutation, 4G 5G mutation. Now, if the mutation causes less activity of inhibitor or more activity of inhibitor, then the coagulation system would change their balance, and that would again lead to many a time thrombophilia. But along with thrombophilia, there is also high risk of developing various misdirection syndrome or miswiring syndrome, and ultimately these people would many a time develop, number one, so-called autism spectrum disorder, and number two, develop psychosis. So these are some of the mutations that give rise to autism spectrum behavior, psychosis behavior due to inappropriate immune system functioning. Coagulation system is also one of the areas of immune system. Now, there is another point. The thing is, our brain has multiple pacemakers. Heart has one pacemaker or two pacemakers. Our brain has multiple pacemakers. Now, these multiple pacemakers many a time has disturbed rhythm because of mutation in various ion channels, and due to that the connections between brain areas are either inappropriately activated or they are not adequately activated. Both the thing would give rise to hyperactivation in some areas of brain. This hyperactivation would again in turn produce chances of having psychosis like picture in patients in some point of their life. Then in molecular front, there is, now we are going into extracellular molecular front to intracellular molecular front. In intracellular molecular front, when we talk about intercellular molecules, we talk about intercellular signal transduction system, or the so-called second messenger, third messenger system. Now in second messenger system, there are various kinases. These kinases generally activate various cellular pathways. But if these kinases are inappropriately mutated and are not adequately activated or more than adequately activated, inappropriately activated, then both the situation would lead to hyperactivity in various cellular signal transduction pathway and hyperactivity in various ways. That would cause destabilization, and those destabilizations would again lead to hyperexcitability of cell and would give rise to psychosis like picture in vulnerable people in some areas. Then comes the molecular surveillance pathway, and this is where psychosis and cancer starts to merge. Many of our patients develop cancer. We don't know why, but having a lifetime diagnosis of schizophrenia or having a lifetime diagnosis of bipolar at any point of life gives rise to high chances of having cancer. Why? Nobody of us can say clearly, but I can give some explanation in this area. For example, I was talking about MyD88 pathways when I was talking about cellular brain normal activity. MyD88 is one of the cancer markers, and its mutation and abnormal activity is linked to a lot of solid tissue cancer activity in body. Now, MyD88 abnormalities are common in psychosis, they are common in carcinoma. Similarly, the BRCA1 gene, which is well-known associated gene of breast carcinoma, as well as ovarian cancer, as well as uterine cancer in some, this BRCA1 gene is associated with various neural connections, various neuronal activity, and it is one of the genes that is of paramount importance during intrauterine brain growth. So, its abnormality would give rise to various psychiatric problems, including psychosis, including bipolar, including recurrent depression, and again it would give rise to pancreatic cancer, breast cancer, and ovarian cancer. That is how they are linked. Then the cellular surveillance pathway I was talking about, cellular surveillance occur at 3 levels. At DNA level, where DNA integrity is being checked by a lot of molecules. Then at intracellular level, where cellular abnormal activity is detected by a lot of pathways, including P53 pathways, and at immune surveillance level. All three areas are associated with a lot of genes. If you go into recent genome-wide association studies of psychosis, which are listed in Schizophrenia Genetic Consortium papers, you would find that many of these genes are these surveillance genes. Many of the genes which are hit in genome-wide association study of Schizophrenia Genetic Consortium or Bipolar Genetic Consortium, they are functioning as this type of molecular surveillance genes inside the neuronal cell and other cell. So we are slowly progressing to a point where we can understand that there is common molecular pathophysiology involved in genesis of malignancy and genesis of psychiatric symptom. That is why many of our drugs are very good drugs for prevention of malignancy. For example, the older generation anti-psychosis drug like chlorpromazine, phenothiazine group, they have associated malignancy reduction chance of around 15 to 20% in large scale Taiwanese study. Similarly, many of the older antidepressants and newer antidepressants are found to be effective against stem cell changing to cancerous stem cell, as well as effective in preventing cancer, because at some point in molecular pathogenesis, the cancer formation, malignancy formation network, gene network, and psychiatric symptom formation gene network intermingle, interconnect, and many times become the same. So these are the ways molecular pathogenesis of psychosis occurs. I have given you few names, few points, and let me be very clear, this is not an all exhaustive list. There are probably at least 10 more I can immediately talk about. I am not talking about because they are still not proven enough, proven adequately enough. I would say not proven enough. There are hints, but I would not talk about anything that is not yet directly referenced by multiple papers. So I am not talking about around 10 mechanisms, and the so far as much mechanism we talked about, we probably don't know more than few folds of those mechanisms. As our science progresses, as our knowledge in science progresses, we would start finding them more and more in future. So molecular neuroscience of psychosis is a fascinating subject.
Thank you for that. I'll give you a chance to catch a breath and take a sip of water if you want while I summarize this section of our talk. That is the molecular side of things. One working knowledge that we need to know as far as the cellular side of things, normal physiology is concerned, is that for every cell there will be three types of mechanisms. One will be pro-growth, one will be one limiting growth, and the third will be towards programmed cell activation, when the limited growth becomes much more operational and you are either talking about recycling the cell or cell death. These then cause various different pathologies can happen with these three variables when we play around with them to cause the preponderance to have psychosis. Essentially, the first thing is that these neurons will be very prone to hyperactivation or chronic excitotoxicity, which I'd like to think that constant depolarization or long-term depolarization would have problems with exhaustion of the resources as far as that neurons is concerned, leading to early cell death. Now, this can happen because of the genetic load that an individual carries inherited, or due to the somatic mutations which happen during the lifetime or at a particular lifetime. This can also happen because of micro neuroanatomical defects in the form of, say, decreased strength of networks or pathways. This can also happen in the form of reduced glial support or support of all the other cells which form a very integral part of our nervous system, like forming the cells responsible for formation of the myelin sheath or the astrocytes, etc. It can also happen because of immune system problems, especially with the innate immune system problems, which causes to destabilization, leading to increased or reduced activation of this immune system, like an example of, say, Brugada syndrome, where increased QT problems with the potassium sodium channels will lead to inappropriate activation, leading to cytotoxicity, erratic brain activation, causing increased chances of psychosis. Can also happen because of the irregulation or dysregulation of the multiple pacemakers in the brain. Intracellular causes for the same could be problems in signal transmission through secondary or tertiary or the messengers. It can also happen because of cellular surveillance pathway problems, which will lead to also similarities with the whole psychosis and cancer side of things, and a hypothesis as to why antipsychotics or older generation antidepressants are also being looked into or have shown some evidence in clinical trials as far as reducing the severity of malignancies or preventing malignancies is concerned. Is this so far a fair summarization of
Yes.
this section of the... Well, Dr. Bhaskar, more than happy for you to grab a drink or take a water break if you
Yes.
want, and then we...
I have already taken the water break while you were
Perfect.
summarizing.
Fabulous. I'd love for us to go slightly more into the depth of the thing that we sort of put a pin on as far as when we were discussing the normal function of the brains is concerned. And we discussed that there would be early signs, especially in cases of OCD or other illnesses. While we discussed that it wasn't within the purview of this topic that we're discussing today, but I think that sort of gave me a lot of interest into. If we want to deep dive into that a little bit, I would love for you to expand on that and tell us a bit more about that.
The thing is, we have to first understand this is a can of worm we are going to open, because this is where we go against DSM, we go against ICD, and we directly say that DSM and ICD is statistics based system and is not clinically relevant. It is only it is actually, I would say, best described as a system which was devised to be used between one clinician to other to say this thing I am seeing in this patient and nothing more. But it has grown into something that has created a lot of problem in a lot of patients' life. Because schizophrenia, the disease doesn't exist. Schizophrenia, the syndrome even doesn't exist. Similarly, OCD, the disease doesn't exist. OCD, the syndrome even doesn't exist, because what we are talking about 3 to 4 billion diseases presenting in various symptoms throughout the life. What is cross-sectionally seen as OCD today would be seen cross-sectionally in 2 to 3 years onward as bipolar. Then cross-sectionally another 5 or 10 years after, the same patient would be seen by someone as schizophrenia, and ultimately the person would get a tag of either schizo-obsessive or schizo-affective, which is again meaning nothing. And so when we talk about these traits, we have to go beyond the DSM and ICD, and we have to talk about that DSM ICD has actually overshadowed and overgrown than what is required in psychiatry, and they are now starting to impose on the growth of psychiatry as a scientific discipline. So when we talk about these things here, then from now onwards, what we are going to describe are lifelong traits, and these lifelong traits are defined by underlying connectomic and genetic configuration of brain. Now, obsessive compulsive personality trait is a trait that starts in utero, that progresses throughout the life, and that stays with patient till they die. This brain trait is neither abnormal, neither this brain trait is disadvantageous. This brain trait has its advantages, its disadvantages. By being by having obsessive compulsive personality trait, our brain has survival advantage in areas where it is threatened. So, in in areas of professional life, this brain would excel. In areas of social life, this brain would excel because there it is dealing with threat efficiently. But this same brain would not be able to relax in known and home environment, and would not be able to deal with home environment or known environment properly, because it is mostly concerned with threat and effective neutralization of threat. So there this brain would suffer various problems. There is no attention deficit hyperactivity disorder. There is again attention deficit trait that would be present throughout the life, and that has neither disadvantage nor advantage. This has advantage in some areas of life, disadvantage in some areas. These people are very good at communication areas. These people are very good in public relationship. These people are very good in social relationship. But these people are neither good enough in a single goal-directed work, neither these people are good enough in maintaining a stable commitment to anything. That doesn't make them good or bad. That just make them something that is innately genetic and that has been with them throughout the life. If we don't think in this way, there would be problem, and we would be randomly diagnosing adult attention deficit. We would be randomly diagnosing various attention deficit persons, whereas it is actually not disadvantageous or advantageous. That is it. Let me give some historical example. Attention deficit trait has always been more in people who resist social indoctrination, who goes out and experiment, and who do a lot of things that are considered taboo by society, because having attention deficit trait makes the person rebel against monotony. So if there was no attention deficit trait in a population, then there would never be any exploration, there would never be any discovery, there would never be anything that has created various change of human civilization. We as a civilization owe a lot of things to attention deficit people. David Livingstone, Scott, Amundsen, they are attention deficit people as well as they are pioneer discoverers. All the so-called pioneers of Western civilization's exploration into New World or expansion into India and other areas to create their empire were done by people who were attention deficit, who were rebellious and who were various kind of pirates in their time. Be it Vasco da Gama, be it Magellan, be it Christopher Columbus, be it Francis Pizarro, be it people who destroyed Aztec civilization, all of them were attention deficit people, criminals, but credited with discovering various things. So we have to understand attention deficit, obsessive compulsive trait, all are traits, and they are present in a fair amount of people, and stamping these people as OCD or stamping these people as ADHD would not solve the problem. We have to understand where these traits are becoming more and creating problem, and we have to understand where the traits are in the beneficial level. We are not yet ready technologically to explore these traits enough, but the solution is not stamping them. So with this, let's go into obsessive trait. Obsessive trait means these people are detail oriented, these people are very methodical, they create a plan, stick to it, they are conscientious people, they are honorable people, but they are that because their brain is hyper vigilant. And why their brain is hyper vigilant? Because their brain has less functional capacity to adjust to different areas. So these are vulnerable people. These are people who has a genetic vulnerability that makes them having less brain functional reserve. So whenever they face various challenges like grief, like prolonged illness, like serious illness, they have more chances of going into so-called prolonged illness behavior or depression. They have more chances of going into various destabilizations. Sometimes the destabilization would manifest as irritable bowel syndrome. Sometimes that destabilization would manifest as panic attacks. Sometimes that destabilization would manifest as various phobias. Sometimes those destabilization would manifest as recurrent low mood, inexplicable low mood, and obsessive rumination. But this is happening because of the same thing that gave them their age in their professional life. So obsessive people are vulnerable people in certain ways, and their vulnerability can be gauged by their lifelong prevalent obsessive trait. The more obsessive a person is, the more structured and the more finicky a person is in their day-to-day life, the more vulnerability, genetic vulnerability they are carrying, and the more chances of their brain going into the prolonged destabilizations is there. So that is what I was talking about when I say that there are signs. So if a prolonged illness has happened to an obsessive person, it is better for the person to be aware that that person has more risk of their physiological illness behavior getting converted into pathological prolonged period of low mood, self-doubt, self-blame, brooding, and various biological symptom, means various other organ dysfunction. If this person has suffered a setback in life, it is more likely than others that this person is going into a severe destabilization syndrome. That doesn't mean these persons are less efficient. That doesn't mean these persons are not reliable, or these persons are less than what others are. that is not. Their brain is actually overcompensating in normal time, and they are more efficient, more productive, more competent than others in normal period. But due to their carry of vulnerability, they have more chance of destabilization than others.
That's a very fascinating,
Yes.
I think, territory that we're walking into as far as the variance side of things. And I think we're also, I think, like you said, it's not just the neurobiology side of things that we're talking about. We're talking about very pertinent, I think, socio-cultural side of things as well when we go into the depths of issues like this. And the idea that normal variance should not only be tolerated but also rather encouraged or celebrated, with the idea that it's what makes us human.
Yes.
And that's why the mind is the most fascinating part of the body, that there are so many normal variations. And the idea that just categorizing it into, say, just like you say, that the blood pressure within this range to this range is normal, we unfortunately probably might need to make peace with the fact and celebrate the fact that you wouldn't have that with the mind. The definition of normality would vary much more than what it would for a mind. It would be much more variable than what it would be for your blood pressure or for your blood sugar. And that's what makes us probably social beings, which helps us evolve more and more generation on generation as well. I would love for you to throw some light on what your thoughts are with regards to emotionally unstable personality disorder or borderline personality, and why does it keep remaining a conundrum when, like, to a very large extent, we are able to provide quality of life to someone from suffering with schizophrenia or someone suffering with bipolar. But in whatever little experience I have in psychiatry, I feel like emotionally unstable personality disorder or borderline personality disorder, either we haven't understood it right, we're not managing it right, something is not right there.
Exactly.
The quality of life that we are still able to give, which is, of course, it's not brilliant. People with schizophrenia, people with bipolar, there is so much more work still to be done. Like you say, we're only scratching the surface. But I feel like, as a science, are we really struggling with EUPD or borderline? I would love for you to share your thoughts on it, go into as much depth as you feel, and for us to have that discussion around borderline personality.
Grinker, Roy Grinker. I don't know how much he is known about in various circles. He would have been happy. Because the thing is, we are dealing with EUPD in a very bad sense of way. We are not treating the things right. Why? Because we have created a EUPD box, which is a garbage box. We throw everything that doesn't matches to our ICD-DSM into EUPD, and we keep it there. This has created all the problem, because we have to understand what is EUPD, what is known by that is not even clear to the people who are diagnosing it, and it is not at all clear to the people who are being labeled by this stamp. There are various ways one reaches to this category, and let me describe them one by one. One is high functioning autism spectrum people. High functioning autism spectrum people, many a time, they are both very obsessive as well as they have a innate attention deficit trait, which is also very strong, and they cannot connect with people. But due to their high functioning nature and their superficial attention deficit trait, many a time this inability to connect with people gets overlooked. They grow up, as they grow up, their superficial charm, which was given by the attention deficit, and their ability to superficially talk with people, gets overwhelmed, and the background core trait where they don't connect socially with people, they don't understand social behavior, that comes out. And ultimately, they reach a state where they are not able to maintain social connection with anyone, they are not able to concentrate on anything, they are not able to meaningfully do anything in their life, and they go into impulsive decisions, they go into impulsive behavior, they go into various compulsive drug use and various compulsive behavior, and ultimately they get a level of EUPD. This is one way a person reaches to EUPD garbage. Then there is another way. This is a simple attention deficit child, but who has intelligence enough to cover it. And mostly girls are actually clustered here, because in girls attention deficit more is expressed via inattention to do any work or inability to do any work sustainably rather than aggression and hyperactivity. So they frequently miss the net, and they grow up being rebellious, they grow up being someone who cannot manage to do things properly, grow up in a clumsy individual, grow up in an individual who gets into fight with everyone, who is rebellious, who is into experiments, who then discovers either cocaine or crystal meth or another stimulant or something like that, and go into various experimental relationship, various sexual misadventures or adventures, and gets a stamp of borderline personality or EUPD. Then there is a third way of reaching there. That is, these are people who have actually high intelligence. Unfortunately, if you ever go into Mensa or 99 percentile or various high IQ societies and talk with their people, you would find out more than their share of people who are termed as EUPD, because this is another way of reaching the EUPD garbage box. High IQ, these people are very high, have very high brain processing speed. They process things very fast. They understand things very fast. So when they are into lower grades, they are able to manage to charm everyone. But as they grow older and older, their ability to comprehend things and their ability to do things also increases exponentially, and they get bored by usual study. Most of them would start experimenting outside the normal school syllabus. They would go into finding things, they would go into debates with teacher, they would go into arguments with teachers, and they would start getting labeled as disruptive kids. Later, they would start finding a thrill in that behavior, they would start enjoying that behavior and enjoying that attention, and many of them in later life would go into this kind of impulsive behavior. There is another side of having exceptional level of processing power. These people cannot connect with normal people easily, because they process things so fast and they understand things from multiple point of view, so they find it hard to connect with people. And again, this makes them slightly less able to connect with people, and if their social sense is not that well developed, or their social understanding is not well developed, then they would again go into either misusing these traits or go into overusing these traits, leading to an individual who is at odds with society, who doesn't understand social norms, who is very argumentative, who is very disruptive, who is experimenting various things and doesn't comprehend social do's and don't. Again, a perfect way to get stamped as EUPD. Then there are people who have opposite end of processor speed. That is, they are having less than normal processing speed. Many a time, they are missed by LD services. They are missed by the less, or if we maintain wokeness and say specially abled service, and ultimately these people would end up being in the same category as their high processing kind peers, and ultimately they would be labeled as EUPD. So there are various ways through which one can reach the garbage box of EUPD. Since it is so varied, there is no one size fits all strategy, and unfortunately when we diagnose EUPD, we find the one size fits all solution. We try to give them one DBT session, we try to give them some psychosocial support, we try to get negatively predicting, we try to actually we go into more kind of negative prediction in them, that this patient is not going to get okay, this patient is not going to get a productive life, this patient would be forever be dependent on social services and things like that, and we start developing a therapeutic nihilism, and that adds to the burden of it. Instead, whenever we get a patient of EUPD, first we have to understand where is this dysfunction that has led to it. We first have to find a core, whether it is the autistic core, whether it is the attention deficit core, whether it is the high intelligence core, whether it is the low intelligence core, where whether, am I audible?
Yeah, absolutely. Whether it is high intelligence, low intelligence,
and whether it is something else. Many a times you would be astonished to know those who are being diagnosed as EUPD today would develop various mitochondrial myopathies tomorrow, various metabolic disorders tomorrow, and I have seen many EUPD ending up as either missed congenital disorder mild variant, or as missed mitochondrial disorder mild variant, or missed genetic syndromes mild variant. So that is another thing that we frequently miss. So we have to first understand what we are dealing with, then we have to create a comprehensive treatment plan where there would be equal importance to pharmacology, where there would be equal importance to diagnosing and if needed up to molecular diagnostic test, and where there would be proper psychosocial support and proper rehabilitation. We should not be nihilistic about them, because in my personal life, in my personal practice, I have seen many EUPD patients getting rehabilitated with adequate pharmacological and other support. And we have to stop banning drugs. We have to stop making these assumptions that only what is written in BNF or various other uh, pharmacology manual is the gold standard. We have to have a free rein in treating them. Many of the EUPD patients respond extremely well to unconventional medication like naltrexone, unconventional medication like clonidine, unconventional medication like stimulants, which we normally don't see in EUPD patients because we don't see them as missed ADHD patient or ADD patient. We don't see them missed ASD patient. We don't see them as missed highly intelligent people who got destabilized. So we don't usually try the treatments that are reserved for this category. We have to remove this ban. We have to understand that psychiatry follows evidence-based medicine only in form, not in reality. Evidence-based medicine means we have to have enough data to formulate a guideline. We have to have enough data to say this thing works, this thing doesn't work. All over psychiatry, if I pull every patient data, still it would not cross a million. Every patient data listed everywhere would not cross million. And standing there with so few data, where most of our studies contain only 100 or 1000 of people, we are deciding lives of more than 8 billion people on this earth. This is not evidence-based medicine. This is not evidence-based at all. This is just another form of eminence-based. We are talking about some leaders who are publishing consensus statements of theirs based on very limited data, and based on that we are treating a Maori in New Zealand, a Bushman in South Africa, and an Aboriginal in Asia without any of them having any representation in any of the data. This is not evidence-based medicine. This is just following evidence-based medicine in form but not in nature. This we psychiatrists have to understand all over the world. We are not following science. We are just following the form. I have one teacher whom I respect above all. I have never met him. He had died many years ago before my date. But Richard Feynman, I would always respect as my teacher. And what he said stands true still today. He said that these subjects don't follow science. They follow the form, but they don't follow the spirit of science. They don't follow the checks and balance of science, and they don't follow the rigorous methodology needed for science. That is still true. Psychiatry doesn't follow the rigorous methodology. We cannot fool ourselves. First rule of science is we must stay true to ourselves. I have to admit that I know nothing. In the greater sense of brain, we psychiatrists know very little. We need to know more. We need to be true to our dedication to brain. We need to have unbiased opinion of our ignorance, and we have to accept that we have to know more, we have to do more. And we should not be following only forms without going into depth of it. I know I have told a very controversial thing, but unfortunately we have to, someone has to ring the bell.
I think it is a very, very poignant and a very necessary statement, especially by someone like you, Dr. Bhaskar, who is so very looked up to. And the idea that anyone who's starting off in the field of mental health or psychiatry or getting to know this subject, I think the fact that we've made this point is very important, because it comes from a place of humility, it comes from a place of modesty, and it comes from the idea that there is so much work to be done. And I think that's what makes our field immensely fascinating, that someone we can talk for almost 2 hours. We've talked about the neurobiology side of things, and probably the concluding remark is we're barely scratching the surface.
Yes.
And that's what makes the brain immensely interesting, immensely fascinating. I think it's been a fascinating conversation today. I would love to have you again on the podcast sometime to discuss further. And thank you so much for being so generous with your time, with your ideas. Thank you so much. I really look forward to having you again.
Thank you for having me, and thank you for giving me a platform to talk about the science in psychiatry. Science in psychiatry is so nascent, so small, that we need to protect it and nurture it and make it grow. Ultimately, if science in psychiatry grows, we grow as a professional, and the people who are getting benefited would be all humanity, because ultimately psychiatry is the subject of brain, and brain is the master organ of body. We are nothing but two eyes, one brain, one spinal cord, and that is what we are. So that needs to be understood by us, that needs to be followed by us. With this, I again thank you for having me.
Fabulous. Thank you so much. I look forward to having you again. Thank you.
Thank you. Bye bye.